Hello, class. I hope you're doing fine. Chapter 8, it's about bacteria and viruses, anything that cause infection, prions, the mad cow disease. And of course, the amount of information is a lot, but I have to summarize it for you guys a little bit. And that's what, I'm not going to go into great detail. This chapter should not take more than half an hour or so. But there are a few important things that you should know, especially with COVID-19 right now. Let's go over it. Okay, bacteria and viruses, of course. This goes back to last chapter, but I thought I'd bring it up, and now never is too late. But the material in this chapter is also going to be in exam number 2. So I brought it up here. It should go with last chapter, but same thing. So what happens when a virus or a bacteria or a fungi or a parasite, anything that enter your body, inside of your body, have the innate immunity, the mucus, the skin, the acid of stomach, the tears in your eye, all of that we studied in the last chapter. But when something can enter inside of the body, an antigen right here, and it is introduced to an antigen presenting macrophage, okay? And then the antigen presenting macrophage make a molecule called interleukin, and the interleukin will stimulate IL, and I spell it out for you here. IL, interleukin, stimulates another T cells, another T cells, another T cells, until eventually you get to plasma cell, and the plasma cell differentiate to memory cell and B cells. So B cells will make the antibodies right here. The antibody attach to the antigen right here. Remember that? So when they attach the antibody attached to the antigen, then another cell, phagocytic macrophages, will engulf that virus or bacteria, what have you, with the antibodies. Then you get rid of the infection. So that is the humoral immune response. Of course, as I said before, in conjunction, they go together with cellular immune response right here. So they are kind of, they go together. Then bacteria and viruses, microbes, microorganisms, they're very small, such as bacteria, viruses, protista, and a few more fungi. Some fungi can be microscopic and cause problems. But the distributed environment and inanimate objects and living things, they are called, I'm forgetting the name. The inanimate object is called the, anyhow, it will come to me and I will say it. Many are useful to humans, and but some bacteria are great for the cheese you eat, the yogurt you eat, the bread you make, the beer, wine. They all are because of the bacteria. So some of the drugs are produced by bacteria. The insulin that we inject to ourselves, people who are diabetics, they are made by bacteria, okay? And that is something we'll discuss in Biology 1, if you take that. And then, of course, what is this one? Without the activity of decomposers of the biospheres, they're also decomposers of the biosphere. They decompose the the organic molecules. Pathogens, that's what the focus of this chapter is, not the beneficial bacteria. The bacteria in our gut, they're beneficial. We have a good amount of them. We have bacteria in our skin. We have a good amount of them. There were studies that thousands and thousands of bacteria within just this body, which they do not harm you and they're beneficial. But the focus of this chapter is about the harmful ones. Okay, they're causing this agent, bacteria, viruses, and others. Defense against pathogens, we talked about these in the last chapter. I'm not going to talk about it. Acquired defense is something that your body that's at the beginning of this chapter. I'll talk about that. That is an acquired event. So bacteria are single cell organisms. Let me see if I can move this. I don't know if you guys can see this or not. Okay, I guess. Okay. So bacteria are single cell. They're prokaryotes. There are three main shapes. The coccus are round one, and then the bacillus are they're like a rod-like looking, and the spirulum one they are curved. Okay, so there are three main shapes are coccus, bacillus, spirulum, and then of course they can be in chain or they can be in clusters. There's a whole bunch of them, which I guess your textbook did not go over into it. That is fine. Most have cell wall, something that plants have, and of course underneath of that is cell membrane. Cell wall contains peptidoglycans, which is a disaccharides with amino acids attached to it. Glyco, it means come from glucose, and peptide, it means amino acids. You already know that. So when the glucose molecules attach to the protein molecules, it's called peptidoglycans. Some antibiotics, such as penicillin, which is taken from fungi, penicillin is a fungi, interfere with the production of the cell wall. So that's why they die, they kill them. Some are surrounded by capsule that allows bacteria to stick the surface of the and protect themselves, and the capsules makes bacteria kind of cohesive, they attach. Okay. Okay, bacteria continued. Bacteria is classified in different forms. If you take microbiology here at Delta College, you will be staining them gram positive, gram negative, and gram positive, their peptidoglycan stains purple, and the gram negative ones, they stain pink. And the bacteria have outer membrane with lipopolysaccharides, which is released as cells, kill the immune system and stimulates inflammation and fever. Here's a bacteria. So very simple organisms. Here's a coccus one. Here is the bacillus one, and here is the spirulum one, the spy one, and this one has flagellum. Not all bacteria have flagellum. Okay, not all bacteria have capsule. Not all bacteria have, but they all have, the DNA ribosomes are needed, which is called in this not only here. Cell wall, cell membrane, and plasmid, not all have them. Most of them have them. Pilus, not all of them have them. Some of them have them. Fimbriae, as far as I know, most all of them have them. Plasmid is a circular DNA in bacteria, which is right here. It's shown to you. Plasmid is circular DNA. So motile bacteria with an appendages of long flagellum, fimbriae, as I said, stiff, adhere to the surface of the host cells, and pilus is a hair-like looking, pili is plural of pilus. I'm not going to read these pili right here. So the genes that bacteria resistant to antibiotic can pass from pilus of the conjugations. So now we'll talk about conjugation here in a minute. Well, let's talk about it right now. When two bacteria come together and they exchange nuclear material, they exchange DNA, that's called conjugation. And then when a bacteria divides in half like this, then that is called binary fission. I don't know where that is going to come up. But plasmid, okay, binary fission, again, I'll talk about that. They are separate. Here is a good example of a binary fission, and so on and so forth. So these are all of the steps in binary fission. I would like you to read them and know them. I cut a lot of information out of this chapter, but these are some of the things I would like to stick with them. Viruses, they're very small. They're smaller than bacteria. I think there is a picture coming up here. They're non-living. We do not consider viruses as living organisms because they do not have the way of metabolism that we have. They do not have it. They do multiply just like us, but you will see their multiplication mechanism here in a minute. Must reproduce inside of a host cell, two parts, an outer protein coat called capsid. anyhow, and nucleic acids inside. So virus is what he's saying. Outside is a protein code, and inside it's either they have RNA or DNA. DNA, as you know, is double-stranded. RNA is single-stranded. And then hopefully we'll talk about both of them in here. Carry genetic information needed to reproduce. Here's giving you an eukaryotic cell right here, an eukaryotic cell. Here's a prokaryotic cell, size-wise, and here's a virus. So you can tell the difference of the size between these three. Viruses continue. In contrast to cellular organism, viral material can be DNA or RNA. I talked about that. Cellular parasites, commandeer and metabolic machinery of a host cell. So let's talk about this. Okay, so this is a virus that, of course, it has a hexagon-like shape. It's not hexagon, but there is a protein molecule capsid is outside, and here are the fiber protein molecules right here, which sometimes they are glycoproteins in some cases. So carbohydrates can attach to these protein molecules. Inside could be DNA or RNA. Again, I'm giving a general picture of a virus. Viruses concluded portions of the virus bind to receptors on the host cell. You will see that once the virus attaches its DNA or RNA enters the cells and codes for proteins. Of course, the virus may have a gene that codes for enzymes needed for the virus and reproduce and exit all cells. The virus relies on the host cells and ribosomes. So right here, another virus, that influenza virus, for example, it has the RNA, it does not have DNA, and they have these spikes, and they have the protein code, the capsid, and they have envelope. So not only they have the capsid, they have another envelope outside of them, which is phospholipid. Capsid is primarily protein molecules. Okay, this is the two ways that viruses multiply. And I'm taking a bacteria cell, which is, it could be a eukaryotic cell, but here they're showing you a bacterial cell. What happens when a virus, this is a phage, bacteriophage, sits on a bacteria? Again, this applies to the eukaryotic cells too, okay? The lytic, I would like you to know, your textbook did not mention this, but I would like you to know the lytic cycle and lysogenic cycle. I would like you to know those. So let's go over the lytic cycle. What happens during lytic cycle? As you can see, it can go either or. So during lytic cycle, the virus sits on a bacteria, it releases its DNA, in this case it's DNA, and then when DNA goes inside of the bacteria, it can infuses itself to the DNA of the bacteria. So this is the blue, is DNA of the virus. It can infuses itself to the DNA of bacteria. And then inside of the bacteria uses the machinery of the bacteria, like ribosomes and other protein molecules, whatever is needed for the bacteria, or the virus, to be reproduced. So the virus reproduce and reproduce and reproduce and reproduce inside of the bacterial cells. And eventually the bacterial cells die and the virus gets out and go find another bacteria to go ahead and kill, okay? Having said that, that is called lytic pathway. Having said that, sometimes when the virus goes and infect a bacteria, release its DNA into a bacteria, then the bacteria multiplies and does not die, but it incorporates the DNA of the virus right here into its DNA, the plasmid. Okay, so the bacteria is multiplying, the DNA of the virus is multiplying too. Can you guys see that right here? Okay, so the virus is multiplying and multiplying and multiplying too. And somewhere along the line of this cycle, the lysogenic cycle, the virus decides that, okay, that's enough. I had enough of my copies, my DNA copies. I'm going to go ahead and make my whole entire self. Okay, so when it makes this whole entire self, starts using the machinery, the ribosomes, protein molecules that the bacteria has, and multiply itself. Of course, the bacteria cells will die. Okay. In this case, the bacterial cells in lysogenic cycle, the bacterial cells do not die. As you can see, the life cycle, the life goes on and on in the bacteria, but in the lytic pathway, they die. Important for you guys to know. Prions, what prions are, they're just a glob of protein molecules. They do not have DNA, okay? And how do they multiply? Still on the question, on the research, a lot of people are working on it. So I'm done with viruses, moving on to prions. Prions, infectious proteins, a few diseases you've heard about, like mad cow disease, spongiform encephalopathy, BSE in cattle, and of course in human. A few other ones, the critical Jacob disease, they're caused by prions. They are just globs of protein molecules that they go ahead and multiply and multiply. Of course, they destroy the host cell as well. Okay, so infectious diseases classified as epidemics. Epidemics, it means you have the disease in an area. Pandemic, it means you have the outbreak, the disease all over the planet Earth. Pan, it means all over. That should be in your prefix and suffix, the root words. It is in there. So here we go. They're talking about Ebola virus. It's an epidemic. It is just right in that Eastern Africa. It can be transmitted to. It did not become a pandemic, unlike coronavirus, which I will talk about that. HIV, it did become a pandemic, but it's not as devastating as nowadays of coronavirus. So HIV stands for, AIDS stands for acquired immunodeficiency syndrome, and HIV stands for human immunodeficiency virus. Okay, so some people can have HIV, but they do not have AIDS. It means they did not develop that disease. It means they have the virus, but the virus is not killing a good number of their white blood cells. Okay, so they do not have developed AIDS. The white blood cells, normal count, they're normal, and the person is continuing. Those people can be contagious. They can give the disease to another person, but they have not developed AIDS. Okay, HIV/AIDS continued. HIV infects and destroys helper T cells. I talked about that a little bit. Specifically, those are CD4, CD8s. There are AIDS in what happens when our white blood cells population goes down. Other organisms in our body, like a common cold, it can multiply and, of course, destroy our cells. Toxoplasma gondii is another organism. When we do not have a strong immune system, we do not have a strong military inside of our body, then what happens? HIV does not kill. What happens? Other organisms that our immune system can easily get rid of or control them, they come and destroy and kill the host. Okay, I hope I made some sense. That's what I mean by other opportunistic infections. Other opportunistic infections like rhinoviruses that we catch and catch a cold. Okay, they come and destroy. Toxoplasma gondii there are many of them. Food poisoning. When you get food poisoning, you come back and get rid of it after day 2, 3, 4. It depends. But if you are immunosuppressed, that's what they call it in case of HIV. When the person is immunosuppressed, then the person poisoning or a common cold, they will die. Okay, the exact dates of the first human HIV, we don't know. I'm not going to go into all of these, you guys. It's not that significant. I don't think it's that significant. But HIV does not have DNA. HIV has RNA, okay? And I like to go the life cycle of the HIV with you guys a little bit. There are a few things. Reverse transcriptase, ACE at the end of the word, it means enzyme. So they have this enzyme that they cause reverse of transcription. I didn't talk about it, transcription, translation in here, but briefly I will talk about it when we get there. Integrase, it's again, ACE at the end of the word, it means enzyme, integrates viral DNA into DNA of the host cells, and protease, they are protein molecules, of course, the ACE enzymes and convert newly synthesized viral polypeptide into a functional viral. Here's the structure of HIV. So right in the middle in the capsid, it has the RNA. At the end of the RNA, did they mention the reverse transcriptase? Yeah, well, right here they mentioned it. Okay, there are reverse transcriptase, and of course it has capsid, it has the matrix, it has the integrase, a protein molecule, protease, all of the good structures that comes with GP120 spikes, they are the ligand, if you would, for GP here 120. It's a retrovirus. A retrovirus means, you should know this, that the virus has an RNA. It does not have DNA, it has RNA. So if it has RNA, how does it multiply? I hope it came up here. So here is HIV attachment, stage number 1. It attaches to the cells. Of course, these are the surface, and in the last picture, sorry, in the last, where are you? Right here, they are mentioning again GP120 spikes. So GP120 spikes attach to the receptors of the white blood cells. The white blood cells have these receptors. The rest of our body, the rest of the cells in our body do not have these receptors. So when they attach to them, they release their reverse transcription occur. Of course, the enzyme reverse transcriptase, it means the RNA from the virus uses the DNA of the host cells and synthesize a DNA that is based on the RNA of the virus. That's called reverse transcription. Transcription, it means when RNA is synthesized based on DNA. So DNA is right here, and when the cells makes RNA, it's called transcription. However, when you have RNA and DNA is synthesized based on the RNA, that is called reverse transcription. So right here, you have the double-stranded DNA of the host cell, and then of course the RNA of the virus right here. Based on the RNA of the virus, the DNA of the host cell makes a DNA that is coded for all of these protein molecules of the virus. So eventually, the virus starts using the ribosomes of the host cell, or white blood cells, and then starts making the protein molecules, and eventually the virus leaves the cell and then goes infect another cell. And when they do this, this host cell eventually will die. Okay, and that's what, how does HIV multiply? You know, we have nowadays, you've heard about COVID-19, we have emerging diseases that occur first time in human, and there are theories that we are getting them from animals because we are cutting deep into the forests, and they are transmitting it. We are interacting with animals more, and that's it's a very strong theory that we are getting them. Re-emerging diseases reappear, significant decline in incidence, simple pathogens, Streptococcus, Helicobacter pylori, which cause ulcer, anyhow. So coronavirus, COVID-19, the abbreviation of COVID, if you're wondering what is COVID, stands for coronavirus disease. I like to add the, the I in there, coronavirus infectious disease. It's an infectious disease. It means infectious disease can be transmitted by, from one person to another person or by fomite. That's the word at the beginning of the lecture. Or if I put this pen in my mouth, this is inanimate object, right? But if it has the HIV and I put it in my mouth, then of course I catch the HIV, or not the HIV, the COVID-19. So these are called fomite. I should put it, fomite. Fomite, it means inanimate objects. It was at the beginning of the chapter. Inanimate objects, I said, it would come to me. It came to me at the end of the lecture. But the inanimate objects that they have the pathogens, whatever it is, causes diarrhea, causes, respiratory inflammation, because whatever it is. So not only we can get diseases by fomites, we also can get diseases by putting our own hand. We touch the surface of a doorknob that infected person touched it, and we touch that, and we put it in our mouth, we become infected. We put it in our eyes, we become infected. We put it in our nose, we become infected. So that's why I like to add the infectious term in here. So, but most places, they do not say infectious, they say coronavirus disease. The 19 was the for the year 2019 that it came out. It attacks the respiratory cells, respiratory tract cells, and then of course some digestive cells as well. The full scope of transmission is not known. Are they being transmitted by arthropods, by insects? If you take a blood meal from one person to the next person, we don't know. Are they being transmitted by sex? We don't know. Okay, that's what I mean by this full scope of it, we still do not know. It's a pandemic, it's all over the earth, it's just not in China, okay? So it is all over the earth. If it was just in China, then that would be an epidemic. Okay, I hope I'm making some sense. I'd like you guys to watch this clip. I don't know how much time-wise I'm doing. I promise I keep it under half an hour, but make sure you watch this clip. It's only 2 minutes, guys. It tells you everything you need to know about COVID-19. And I could ask some questions from it. You should know it's an RNA virus. It's not a DNA virus. Viruses either have RNA or DNA. They do not have both, okay? So that's what COVID-19 is. And that's what that clip, 2 minutes clip says. It's wonderful. You should watch it. Here we go, emergent diseases. He's giving you different type of disease all over the world, what happened. And this chapter is not the focus of this. The Zika virus that was famous before coronavirus, it infects babies and then transmitted by Aedes mosquito. So we don't know COVID-19 is being transmitted by any mosquito or not. We just talked about that. Middle East respiratory syndrome, MERS, that's another one that, again, make sure you study whatever is in the PowerPoints. Whatever it is fine. This chapter is huge. I think it gave you too much information on a lot of things, but I would like you to focus for this chapter, focus on what I said. Antibiotic resistance. So if you give, and I have a picture, if you give antibiotic to a person and they do not completely take it, they do not take it for the 10 days or whatever days the doctor prescribed, what happens? Some bacteria inside of the body remain alive. You did not get rid of, you got rid of your symptoms, you got rid of your infections, but there are few of them that stays inside of the person. And if that one is transmitted to the next person, that antibiotic that worked on you is not going to work on that person. So it is very critical that, and then of course, you've heard of tuberculosis. At the beginning when tuberculosis came, and then of course it was always here on planet Earth. But at the beginning when the penicillin came to kill tuberculosis, it was great. It was great that people were cured. But now there is a resistance to it. Okay, right here. So uh, um, here is a not drug resistant, and these are a few of them that drug mutant, drug resistance. So when you give them, you transfer them to another plate and they multiply, most of them that multiply, they are the drug resistance ones. Okay, imagine each one of these are a person. Okay, each one of these is a person. And the story of tuberculosis, well, tuberculosis is a famous one, but most of the diseases that we talked about, they are the infectious diseases that are caused by bacteria. I'm talking about bacteria, focusing on bacteria. The infections, the resistance occurs over a period of time. Okay, so everything I said is right here. Prevention of antibiotic resistance, make sure you take them all. And that would be the end of the chapter. As I said, I'll try to make it short. And then if you guys have any further questions about this chapter, please let me know. And I'll be here to answer your